Hi there. Good afternoon, and thank you so much for joining me. This is Mrs. Henry, and I am here to provide you with a mini lecture on the cell cycle. So let's go ahead and get started. As always, first thing we want to do before we get started with any lecture is make sure that you have a good understanding of what you should know after we are finished with the lecture. So once this mini lecture is over, you should be able to define what the cell cycle is. You should be able to tell me the two major stages of the cell cycle and kind of a brief description about what happens during these stages. You should be able to list the substages of interphase and the mitotic phase. And you should also be able to explain to me why cells stay so small. Even in multicellular organisms like elephants that are very large, usually tons, they weigh tons, their cell size is still really, really small. So we're going to describe why that is. So let's go ahead and get started. So here off to the side here, you see a human life cycle. So we started out as a fertilized egg, a zygote. And then that fertilized egg starts to divide and we create a fetus. And then once the fetus is fully developed, we're born into a baby. And then from a baby, we grow to a child. And then we become a teenager, an adult, old age, and then we all die. So just like the human beings have a life cycle, the cell also has a life cycle, and we call that the cell cycle. So the cell cycle is just basically the life cycle of a cell. And all cells have a cell cycle. However, in this mini podcast, I am just going to be talking about eukaryotic cells and the cell cycle of a eukaryotic cell. Prokaryotic cells also have a cell cycle, but that is not going to be the gist of this lecture today. And so if you've kind of forgotten what's the difference between a prokaryotic cell and a eukaryotic cell, we're going to kind of discuss that again. So a eukaryotic cell, you can see here at the bottom, it has a nucleus and it has membrane-bound organelles like endoplasmic reticulum, the Golgi apparatus, mitochondria, things like that. A prokaryotic cell is much simpler. It may have like a cell wall and a capsule, a cell membrane, some cytoplasm and DNA, but there are no membrane-bound organelles. There's no nucleus. And so generally, prokaryotic cells are archaea and bacteria. They're fairly simplistic. Eukaryotic cells are more complex. So prokaryotes do also have a cell cycle, and they divide by a process called binary fission that we've talked about before. Eukaryotic cells, because they are a little bit more complex, they do have a much more complex cell cycle as well. All right, so off to the side here, you will see a circle with different colors. And on the outside of the circle, you'll notice three gray stages, G1, S, and G2. And then you notice this kind of salmon colored portion over here that's labeled M. So collectively, G1, S, and G2 make up the first stage of the cell cycle, which is called interphase. So if this were like a pie, notice that the majority of the pie is interphase. That is because the majority of the time, cells are in the phase of interphase. And what happens during interphase? Well, during interphase, the cell is going to grow. It's going to carry out normal cell functions. For example, it's going to need to metabolize some sort of energy source to get energy. It's going to need to duplicate its DNA, because if we're going to divide eventually, we're going to need to make sure that each cell has the right amount of DNA. It's going to create proteins. Proteins are important for a plethora of reasons in a cell. But one reason proteins are so important is because they help divide. And it's going to prepare to divide. During the mitotic stage, basically, we are actually going to divide that cell. Okay, what I said, generally, cells spend the most amount of time in interphase, and depending on the type of cell that you have, the cell cycle can be longer or shorter. For example, your hair and fingernails grow relatively quickly, so their cell cycle is shorter. than say like some of your other cells. However, there is one instance where cells spend more time in the mitotic phase, and that is if you have cancer. So while most cells only will divide under certain conditions, so we make sure that we have the right nutrients, we make sure that it's the right time, cancer cells will just continually grow, because really what cancer is just uncontrolled cell growth. And so you'll notice here the normal cells, if there are positive growth factors, in other words, we've gone through checkpoints and everything looks okay, the cells will divide. But if there are negative growth factors, we don't have enough nutrients, maybe we don't have enough light if we're a plant, then the cell's not going to grow. However, if you have cancer, whether or not you have positive or negative growth factors, you just continually divide. All right, now let's get into some of those sub-stages of interphase. So notice there are 3 sub-stages of interphase, G1, S, and G2, and those actually stand for something. So G1 is also known as the gap 1 phase of interphase, and it comes first. And so during that phase, a cell will carry out its normal cell functions like we talked about before. So it'll metabolize things using energy, it'll create proteins, it's going to grow, things like that. Then we get to the S phase of interphase. That is known as the synthesis phase. And if we synthesize something, we're actually making something. And so we're going to make more of our DNA so that we're going to duplicate those chromosomes so that when we actually do divide in the mitotic phase, all our cells have the right amount of DNA. And then during G2, which is up here, also known as gap 2, the cell creates proteins and prepares to divide, because we're about ready to divide here in this mitotic stage. So we need to make proteins, we need to create what's called a spindle apparatus to help us divide. Now you'll notice some arrows off to the side here with this G and this 0 here. This is actually called G-nod. And so during G1, sometimes cells will decide to go into what's called G-nod because they are not going to actively divide. There's a couple different reasons they might do that. Some cells, the conditions aren't right, like we talked about if they're not cancerous cells, but there's not enough light, there's not enough nutrients, something like that, the cells will go into G-nod. So if you think of like plants during the winter time, if it's a deciduous tree, it's not going to create leaves, right? So it'll go into that G-nod where it's not going to actively divide. But some cells, after a certain time period, they automatically go into G-nod and they don't ever divide again. So you'll notice a couple cells over here. This is called a neuron. This is your nerve cells. And we also have a muscle cell over here. So nerve and muscle cells are in G-nod pretty much permanently. So that's why if you have damage to your spinal cord, or you have a brain injury, or you injure a muscle, those cells can't reproduce and fix themselves. So sometimes you unfortunately have permanent damage there. All right, next I'm going to go on to the mitotic phase. So in the mitotic phase, there are 2 substages. Those 2 substages are mitosis and cytokinesis. So mitosis is where just the nucleus is going to divide, and it's actually further broken down into 5 other substages: prophase, prometaphase, metaphase, anaphase, and telophase. And we'll talk more about those in a subsequent video. So right now, I just want you to kind of be able to name them and list them. There is a lot that happens in each of those stages, but we're not going to go into that today. And so an easy way to remember that is kind of if you can remember PMAT for the prophase, and then the prometaphase kind of mixes together, and then the metaphase, anaphase, and telophase. It's an easy mnemonic to be able to remember that. And then during cytokinesis, well, what does cyto kind of look like? That kind of looks like cytoplasm. And so the cytoplasm is dividing with all of those organelles that are going to split in between the 2 cells. All right, last thing we're going to talk about is cell size. So I have a mouse here and I have an elephant here, and it says, which has larger cells, a mouse or an elephant? So instinctually, you would think, well, of course the elephant does, because the elephant's much bigger. But actually, that's not true. They have about the same cell size. It's just that an elephant is made of many, many, many more cells than a mouse is. So why is that? Well, there's three main reasons that a cell remains small. The first reason has to do with the surface area to volume ratio. The second reason is communication. And third reason is transportation. And we're going to get into a little bit more information about that here in a second. All right, so let's talk about this ratio of surface area to volume. So I want you to imagine for a minute that you have a balloon and you start to blow air into the balloon. The outside of the balloon would be the surface area. The inside of the balloon would be the volume, the air that we put into the balloon. Now you can continue to blow more and more and more air into this balloon. But eventually, if you can keep blowing more and more and more and more air into the balloon, what's going to happen? Well, it's going to pop, of course. Why is that? Well, because just like a cell, both the surface area is growing and the volume is growing, but the volume grows at a much faster rate than the surface area does. So if you think of a cell kind of like a cube with 6 sides, to figure out the surface area, we take length times width times 6. But to figure out the volume, we take length times width times height. So that volume grows at a much faster rate than the surface area does. If the volume exceeds the surface area, the cell will pop. So that is why, one reason why cells stay relatively small. What are some other reasons that cells stay relatively small? Well, if you kind of think about like working in a building, for an example, let's say a school, and you want to talk to a teacher, and you have a teacher that's right next door to you, maybe right across the hall. Theoretically, you probably wouldn't do this, but it would be fairly easy if you had to take a paper over to that teacher to walk right across the hall. Or if you really needed that teacher, you had an emergency and you hollered at the teacher's probably going to hear you, if you holler. So the communication is pretty easy. Transportation is pretty easy. Same thing with cells. The smaller the cell is, the easier transportation is. So you kind of see like a little diagram here. This is time over on the y-axis, and here's cell size. And so there's a certain size that cells get to that it makes communication and transportation a lot easier. And so this is called a micrometer. And so you'll notice as the cell gets a lot bigger, communication in both prokaryotes and eukaryotes and transportation is a lot more difficult. You also notice, though, that prokaryotes tend to be smaller than eukaryotes, and that is true. Eukaryotes are a little bit bigger, so they can grow a little bit bigger and make transportation and communication easier. So just kind of to recap, so come back to those objectives, those first objectives that I had at the beginning. You should be able to define what the cell cycle is. So remember that the cell cycle is just a series of stages of the life cycle of a cell. There are 2 major stages of the cell cycle. Remember that they are interphase, and that is what the cell spends most of its time in, and the mitotic stage, which includes mitosis and cytokinesis. That is where the nucleus divides and the cell itself divides. Okay, be able to list the substages of interphase and the mitotic stage. So remember that the substages of interphase were G1 or GAP1, S or synthesis, and G2 or GAP2. Remember, during G1, the cell grows, carries out normal cell functions. During the S phase, we duplicate our chromosomes. And during GAP2, we're getting ready to divide. The substages of the mitotic phase are mitosis and cytokinesis, and the mitosis is further divided into prophase, prometaphase, metaphase, anaphase, and telophase. And finally, describe why cells stay so small, even in multicellular organisms. Remember that they stay small because the surface to volume ratio, volume grows a lot quicker when the cell grows than the surface area does. It provides for easier communication and easier transportation. And I hope that was helpful. And I thank you so much for joining me.